All living organisms construct proteins and enzymes using information encoded in DNA. To regulate the changing needs of the organism, genes along the DNA molecule are expressed or suppressed, depending on the cellular context. Epigenetics examines the complex inputs which chemically modify the structure of chromatin to render it more or less accessible in a heritable manner. For example, a methyl group can be added to or removed from histones, which may silence or activate a gene. Therefore, in the presence of polycomb, genes are silenced, whereas loss of polycomb opens up chromatin and induces transcription. The environmental characteristics navigated by the developing cells is demonstrated by the Waddington landscape, an illustration of how regulatory factors influence developmental paths. These dynamic and adaptive gene responses are a normal part of our physiology and development, but like mutations in the genes themselves, they can also go wrong. The prevailing model of cancer formation emphasizes somatic cell mutations, or mutations in cells which are not eggs or sperm. This somatic mutation theory proposes that a succession of several DNA mutations in a single cell causes cancer. A recent experiment on Drosophila fruit flies explored cancer generation via an entirely epigenetic mechanism, inspired by epigenetic research conducted over the last 2 decades. By temporarily reducing the activity of PRC1, an important epigenetic regulatory protein complex, researchers induced cancer in Drosophila cells. Even when PRC1 activity was restored after 48 hours, the altered cells retained their cancerous traits. This temporary PRC1 depletion triggered the induction of several genes, including signaling components that drive cell proliferation, dedifferentiation, and migration. These genes created a self-sustaining expression which continued even after the PRC1 complex was restored. Therefore, cells with a DNA compatible with normal development can experience a transient perturbation, leading to the formation of a cancer. These findings, along with the contributions of other research groups, warrant a reconsideration of the somatic mutation theory. The fact that initial cancer-causing events might be epigenetic could inspire new research and novel therapeutic strategies.